direct analysis of urine with high-throughput sample deposition and ambient ionization. In these experiments, we have completed rapid analysis by using more accurate control of the sample size and its deposition on our commercially available QuickStrip 96 consumable. We selected the TTP LabTech Mosquito for this task as it is known for its capability to deliver sub-microliter volumes with good accuracy even when sample viscosity information is limited. The Mosquito is programmed to collect a specific volume of sample from each well of the SBS plate, move the QuickStrip into position under the micropipette tips, and eject the sample onto the QuickStrip. By design, the Mosquito performs accurate deposition of sample onto the QuickStrip consumable, which is positioned and adjacent to the sample plate on the Mosquito deck. A new pipette tip is used for each sample deposition. In this case, we are eluting 1 microliter of sample onto each column of the consumable. Once sample deposition is completed, with the QuickStrip sandwiched inside the mounting plates, it's then moved to the DART-equipped mass spectrometer for analysis. For these experiments, we utilized a ThermoQ Exactive. Automated sample analysis occurs as the consumable is moved through the ionization region at a user-selected speed depending on the experiment. For the purpose of demonstrating the utility of the DART HTS for high-throughput screening, we subsequently spiked the urine with increasing concentrations of drugs and introduced an internal standard to permit normalization. Our method is outlined here. Analysis of 200 nanoliters of normal urine with ultra-high resolution mass analysis permits determination of numerous metabolites and waste products by using their accurate mass values. Analysis of 96 samples of urine spotted on 4 rows of the QuickStrip with 384 well spacing is shown using mass chronograms to highlight the presence of each spiked drug and the urea tetramer. Showing one subset of 24 samples, the mass chronogram for protonated cocaine is shown in the top box. Utilizing the mass chronograms, we can make a quick assessment of detection limits for each of the compounds. Here, we move to view the signal from samples containing 100 nanograms per mil of cocaine. Detection is observed, and although the signal appears to be low, it is significant. We show 250 nanograms per mil, as this is the cutoff used for prosecution in some of our customer facilities, 500 nanograms, and 1 microgram exceed the cutoff and are clearly strong signals. Looking at the bottom trace for the urea tetramer result for the last 4 samples, show that there is no carryover and indeed no cocaine or standard in the blank urine, as is expected. Utilizing the ion intensity yielded by the internal standard, we normalized the data and then produced a standard curve for the cocaine, and also for methadone. The drug-related ions do not stand out in the 250 nanogram per mil mass spectra, which is quite complex. However, zooming in on the mass range of the protonated drugs and their deuterated standards show that they have significant abundance relative to the background. The 15,000 resolution spectra acquired at 3 scans per second are sufficient enough to permit accurate mass confirmation. In conclusion, we see that high-throughput analysis of urine is quick and simple with this system, and therefore enabling rapid, high-throughput drug screening with this method. Contact us for more information at sales@IonSense.com.